Hello everyone, welcome to lesson 1.3. In this lesson, we're going to be covering membrane structure, including going over the functions and actual structure of the plasma membrane and its primary function and other biological and physiological features of it. So let's get into it. So before we actually get into the lesson, there are some vocabulary words that you should be familiar with and know. So let's go over those first. So the first one is hydrophilic, and these are substances that are attracted to water. The second one is hydrophobic. These are substances that are not attracted to water and are repelled by it. And there's empapathic. And empapathic substances are substances that are hydrophobic and hydrophilic. So they have both water attractive and water repellent properties to them. All right, so moving on to phospholipids. So all plasma membranes and all cell membranes are composed of phospholipids, which are organic structures that are made and composed of two parts. And the two parts of the molecule are often called the phosphate head, which is hydrophilic, so it's attracted to water, and the two hydrocarbon tails, which are hydrophobic, so they're repelled by water. And together, phospholipids have empapathic properties. So as you can see here, the phosphate head is attracted to water, and excuse me, the hydrocarbon tails are repelled by water. And phospholipids will form a bilayer in water due to the empapathic properties of the phospholipid molecule. So yes, phospholipids have a polar charged phosphate head that is attracted to water, and they will have two long nonpolar lipid hydrocarbon tails that are repelled by the water. And so, because of this empapathic properties, the phospholipid will then orientate itself in which a bilayer is created that only allows certain molecules to actually enter a cell. So when phospholipids are mixed with water, the phosphate heads are attracted to the water and the hydrophobic tails are attracted to each other. And then this will form a bilayer. The primary function of the phospholipid bilayer is to form a barrier through which ions and hydrophilic molecules cannot easily pass. Individual phospholipids can actually move within the membrane, and this allows for membrane fluidity and flexibility. Membrane proteins. So these are essential structures that are present in all cell membranes, and they are embedded into the phospholipid bilayer. They can be permanently fixed or temporary on the bilayer, and there are two primary types of membrane proteins. There are integral proteins, which are embedded inside the membrane, and these are hydrophobic. And then there are peripheral proteins. These are embedded outside of the membrane, and they're hydrophilic. All right, so membrane proteins have a wide variety of functions. And a manner in which you can remember this is through the abbreviation JITRAP, in that they assist in junctions, which connect and join two cells together. Enzymes, so they help catalyze reactions in the cell. Transport, they're responsible for facilitated diffusion and active transport in the cell. Recognition, they act as markers for cellular identification. Anchorage, they mark attachment points for the cytoskeleton. And transduction, so they function as receptors for peptide hormones in the cell. And here's just a diagram to represent the various functions that membrane proteins employ in the cell membrane. All right, moving on to integral proteins. So these are proteins that are permanently attached to the membrane, and they're typically transmembrane, so they span across the entire bilayer, and they tend to be hydrophobic on some parts of their surface. And their primary function is to transport substances and other materials across the actual membrane. Then we have peripheral proteins. Peripheral proteins are hydrophilic on their surface and are therefore not embedded within the membrane. And they sit on the membrane surfaces, and they can slide around easily and collide with each other, but they cannot flip from one side of the membrane to the other side. Most peripheral proteins are attached to integral proteins, and they are usually involved in maintaining the cell's shape or motility and mobility. They may also be used through enzymes in catalyzing reactions in the actual cytoplasm of the cell. And here is just a diagram that represents integral and peripheral membrane proteins and their primary functions. All right, moving on to cholesterol in membranes. So cholesterol is an essential component of animal cell membranes. The function of cholesterol is to maintain integrity and stability in the cell membrane. Cholesterol is an empathic molecule, so it has both hydrophilic and hydrophobic properties. And cholesterol is a type of steroid, and a steroid is a type of lipid, but it's not a fat or oil. Cholesterol is oftentimes positioned between phospholipids in the actual cell membrane. And so the role of cholesterol in membranes. So cholesterol makes the phospholipids more tightly packed and regulates fluidity and flexibility of the actual membrane. And it keeps the membrane from being too fluid or too solid. And this is why it is sometimes the membrane structure is sometimes referred to as the fluid mosaic model due to the properties of cholesterol in the actual membrane. All right, so the fluid mosaic model of the membrane structure is the primary structure that we use to actually represent and scientifically illustrate what the plasma membrane and actually looks like. And it's referred to as fluid because it can change shape, but also because the phospholipids can change position freely in the same plane. And it's referred to as a mosaic, as the membrane has protein molecules that are embedded and attached to its surface. All right, so moving on to illustrations of the fluid mosaic model of membrane structure. So as you can see here in this following image, this would be a general diagram and scientific illustration of the fluid mosaic model of membrane structure. Whenever you're drawing a scientific illustration of the fluid mosaic model, ensure you are incorporating and including these primary structures as criteria for the drawing. So ensure you're including phospholipids, integral proteins, peripheral proteins, cholesterol, channels for facilitated diffusion in the plasma membrane, and then pumps for active transport in the plasma membrane, among other structures. Models of the membrane structure. So the current model of the membrane structure is referred to as the fluid mosaic model. However, before use of this model was modernized, there was another model that was commonly accepted by scientists and researchers alike, and this was known as the Davison and Danelli model. So, the Davison and Danelli model was a proposed theory and model that the lipid bilayer was coated on either side with a layer of globular proteins in the cell membrane. However, there were several issues that were wrong, several issues with this actual model. For one, it assumed that all membranes were of uniform thickness and would have a constant lipid-protein ratio. It also assumed that all membranes would have asymmetrical internal and external surfaces, and it did not account for the permeability of certain substances in the actual membrane. So, for example, did not recognize the need for hydrophilic pores in the actual membrane. And because of these doubts and issues with the actual model, several pieces of evidence were conducted through experiments, and this evidence was gathered and used against the model. So membrane proteins were discovered to be insoluble and varied in size through experimentation, and so it could not be one straight layer like it was originally thought the membrane was. And then fluorescent antibody tagging of membrane proteins showed mobility in the membrane, and freeze fracturing showed that there were proteins inside the membrane and not just outside. And so here is a visual representation of the two models. On the left, you can see the Davison and Danelli model. We have the lipid bilayer, and either side, excuse me, it's coated in distinct layers with globular proteins. And then on the right, we have the fluid mosaic model. And this is the more modern model. And this is where proteins are embedded within the bilayer and they're not just coated on either side. So that's it for lesson 1.3. Thank you for listening, and I'll see you next time.